and then afterwards she moved to Belgium to work in microbiology and computational biology. So thank you very much, Mireia, for accepting the invitation to present your work here, and we are very much looking forward for your talk.
Thank you. Thank you very much for the invitation. So yeah, as Anna said, I'm going to talk about our recent work on the person-to-person transmission landscape of the gut and oral microbiomes. Okay, first, a short introduction. As I was told that this is the first talk on the microbiome. So indeed, the human microbiome is a collection of microorganisms that reside in and on the human body. We have, therefore, roughly as many bacterial cells as human ones. And in the gastrointestinal tract, that is the most densely populated ecosystem. We have on average 10 to the 12 bacterial cells. So it's, we have really many of them. And it also, I mean, the microbiome is not only important because of its number, but it also plays a key role in maintaining our host health. And alteration in its composition have been linked to a very wide range of different diseases, not only gastrointestinal diseases like Crohn's disease, celiac disease, and so on, but also many other diseases like diabetes, Parkinson's, Alzheimer's, depression, and so on. And so what about transmission? So we know that the human microbiome plays a key role in maintaining our health, and we also start having some good knowledge on what factors modulate its composition. However, we know very little on how we get those microorganisms. So how these health and disease-related microorganisms are spread, so acquired by individuals and spread in populations, which is to us intriguing because for some pathogens, like COVID-19 would be a clear example, in a very little time, we got to know a lot. And in contrast, for the microbiome that has been living for us forever, we just don't know it. So indeed, we think that understanding transmission of the microbiome is not only a fundamental question for microbiome research, but also one with very important implications. As to the microbiome, non-communicable diseases, so those that are not linked to an infectious component, could become communicable. And also, if we understand how this transmission takes place, we would also have the potential to understand and alter the transmission of these microorganisms. And this could eventually lead to improved microbiota modulations, both on the timing, so knowing when the host is most susceptible to these modulations and when they are more effective, but also on the composition, so where there are some strains that are more efficient in actually colonizing the receiving host. So what do we know so far? We know a bit on vertical transmission, which is the one that takes place between mothers and infants. And actually the first colonizers of our gut are maternal microorganisms, followed then by microbial succession processes. And our lab started studying this process of mother to infant transmission some years ago and started developing the tools that would be needed to perform this high resolution strain level analysis. This was done thanks to advances in metagenomics, which is what we need. So we need this very high resolution so that two bacteria into individuals, we have enough power to say, okay, these are the same. If we don't have this high resolution, we could just, this could just be the result of similar diets, for example. But if we perform this very high resolution analysis, then we can build these phylogenetic trees. And when we see that some leaves of this tree are very close together, we can infer that there was a transmission event there. So with this first study, we found something very intriguing, that was that maternal strains were more persistent in the baby gut than other ones, which would hint towards better adaptation of these maternal strains than the environmental ones. But of course, in babies, many of the very typical adult bacteria are missing. So we must get them later on. And that's when we started studying these processes more in detail. So this is work I did still during my PhD in Belgium, where we set up a multi-generational family cohort. So we had 24 families with at least 3 generations of women, so from the grandma to the mother to the daughter, but some even with 5, spanning a very wide age range in the confined geographical areas of Flanders in Belgium. And we profiled these microbiomes at the strain level for 51 species in this case. And we found that transmission also occurred across multiple familial generations. It was not a very frequent process, but we had some observations of that. But when we started to look at this more in detail, we found that kinship, so being part of the same family, indeed played a strong role, but cohabitation even a more important one. So it seemed that the environment, so living in the same house, would also play a role, giving us some hints on horizontal transmission. As indeed, we have some evidence already for a decade that cohabiting individuals, also those that are non-kin, display some microbiota similarities. This, as I was saying, could also be the effect of, for example, a shared diet. But actually, some years ago, horizontal microbiota transmission in households was demonstrated in a small cohort in the Fiji Islands. And also then some hypotheses started to be put forward on what microorganisms could be spread through each mode, and what would be the properties that would allow them to be transmitted. So this was our base for us to set up this large-scale study of microbiome transmission. We profiled there more than 800 species at the strain level. And what was important for us is that as there is still a very large majority of the microbiome that has never been cultured, through metagenomics, we could also have the possibility to study strains of species that have never been cultured. Because, of course, when we extract the DNA from a sample, we just see all the DNA that is there. We don't need to culture these bacteria. So indeed, 55% of the profile gut species and almost 25% of the oral one are so far uncharacterized, have never been cultured. We then tried to put together as many samples as possible. So we included all publicly available data that was there, but also some new data sets that we could obtain through international collaborations. As for us, it was important to try to get a good representation across the globe, trying to reflect different human lifestyles. We then also did some methodological developments to try to infer microbiome transmission in a way that was as precise as possible. And this was based on different properties of strains. So these were being mostly like very person specific. So we know that a same strain cannot, be found in 2 individuals if those are not in contact. That's extremely rare. And also we set up a species specific operational definition of strains based on these properties. So for each of the species, we could build a phylogenetic tree, and then depending on if leaves were close enough or not, we would call them the same strain or not. And if it was the same strain, we would consider there was a transmission event. And so on the results, what did we see? So we saw a gradient that was linked to a sharing environment, which to us was something very interesting. So on the gut microbiome first, which we studied through many stool samples that we included and public data, could see that, of course, 2 samples from the same individual taken at different time points. We had up to 6 months apart, which are the most of strains, 87% on average, followed by mothers with their offspring, but also individuals in the same households or twins or people in the same village, so that would still share an environment, which share a good amount of strains. In contrast, people that were just not in contact in the same or in different populations had a median of a 0% strain sharing rate. So indeed, environments seem to be playing a very strong role. A bit more in detail, we then looked at vertical transmission of the gut microbiome, and we saw that in the first year of life, strain sharing was very high, so 50% as a median and stable. And And then it started decreasing together with the accumulation of strains from other sources. But what was intriguing for us is that even in older individuals, so these were between 50 and 85 years old with their mothers still, those individuals, they would still share more with their mothers than with mothers of other individuals, meaning that vertical transmission is both considerable, but also very long lasting. We also checked then an influence of delivery mode. We know that whether infants are born by vaginal or by C-section plays an important influence on their early life colonization. And so that vaginally delivered strains shared more strains with their mothers as compared to those born with C-section. But actually the difference was vanishing later on. So after 3 years of age, there was no significant differences between the two groups anymore. We then looked at the horizontal transmission of the gut microbiome and saw that between individuals sharing a household and different relationships, so we had mothers with the offspring, father with the offspring, siblings and partners, they all shared comparable amounts of strains, and all of them were significantly higher than those on different households. But there were no significant differences among those. So this also tells us again, okay, environment plays a very strong role, as when these children get older, they do not share more with the mother than with the siblings or with the father anymore. We then also looked at adult twins who did not live apart, who did not live together, anymore. And so that strain sharing among them declined steeply with the years they lived apart, much more so than with their age, meaning that when they stopped living together, their microbiome diverged importantly, and much more so the fading influence of the vertical transmission from the same mother at birth. We also saw a slight impact of genetics here. So monozygotic twins in any age category or years living apart tending to share more strains than the zygotic ones. And we even went one step farther. So then we also check gut microbiome transmission within populations, and so that we could sort of build strain sharing networks that recapitulated population structures. So indeed, through tracking microbiome strain sharing, we can also look at how close people are to each other geographically. Then on the oral microbiome, so all the saliva samples that were analyzed here, we see something different here. So we did not see this impact of mother to infant transmission in early life, which makes sense because of there is not the delivery canal there. But in contrast, there was much more strain sharing in general. So if we're seeing that people living together shared the median 12%, here it's around 30%. And partners tended to share more, even if the difference is not significant, than mothers or fathers with the offspring. And indeed, going now here to the right, we saw this accumulation. So here it was not that offspring with the mothers shared every time less, but it was more. So the more time two people spend together, the more strains they shared. And this was also true for fathers with the offspring and mothers with the offspring. And then also it seemed that in households that there were more strains shared in general, then there were more strains shared across any kind of relationship. We then had another question. So it was, are all the species transmitted equal, or are there some bacteria that are better at being transmitted than others? And we saw that no, there were indeed some bacteria that were transmitted much more often than others, but also strain sharing rates of the bacteria were rather consistent across data sets. So it seemed as if this was a property of the bacteria. We then looked at what species tend to be very often highly vertically transmitted, horizontally transmitted, or in populations. And we saw example for vertically transmitted species that were bifidobacteria, that were typical infant bacteria, but also some other ones like Bacteroidetes or Akkermansia that has been strongly linked to health and disease. And then in households and in populations, there were many of the under-characterized bacteria, those that I was saying in the beginning that have never been cultured, that were amongst those that have never yet been cultured in the lab. And that was similar for the oral species that were highly, so very frequently transmitted. So we saw these were the ones that were highly transmitted in any of the different relationships that we were looking at. And as you can see, some don't even have a complete name, meaning that they have not been characterized, so cultured in the lab yet. And this is calling us for studying these bacteria better. Because we see that some of the more transmissible bacteria, we just don't know them, we don't know anything about them. As a last step, we are then trying to see if there were some phenotypical properties that could explain these transmission dynamics. So if there were some properties that were shared between the bacteria that were transmitted more often. And so that was kind of the case. So in general, properties that made the bacteria more resistant to the environment. Here, for example, it's important that many of the bacteria in our guts are anaerobic, so they just die in the presence of oxygen. So the ones that were aerotolerant, that they could tolerate the oxygen, they tended to be more often transmitted across longer distance ranges, together with those that could form spores, so resistant forms. For the oral microbiome, in contrast, that was not so much the case. We could not link it to very clear properties. But what's interesting here is that in another study, we also assessed in the context of fecal microbiota transplantation, which is another case of transmission. So the one that we were studying here is the result of one bacteria being able to survive in the environment and then being able to colonize a new environment. In FMT, in contrast, we just put the bacteria there, so they just need to colonize. And we could see that were some properties that were shared. So for example, gram-negative bacteria, they were also here engrafting more often in fecal microbiota transplantation. So to finish, as take-home messages, so we showed that the microbiome is transmitted vertically, but also horizontally, and very extensively, more than what we could have thought before, that sharing an environment plays a very important role. So cohabitation was very important here, and that we saw distinct patterns for the gut and for the oral microbiome. So not all the microbiomes are equal, and they are all not transmitted equally. And finally, that it's in the transmission was not left to chance. So there are some bacteria that seem to be dispersal specialists, and tend to be those with certain phenotypical properties that are transmitted more often. So with this, I would like to thank all my colleagues, and please, I'm happy to take any questions. Thank you.
Thank you very much, Mireia, for a fantastic talk. Congrats on the work. So now, as you said, it's time for questions. Anyone who wants to ask a question, now it's the time. I see that Romulo Silva de Oliveira has a question. Maybe he wants to ask a lot.
Hello, good afternoon. Thank you so much for the opportunity. Congrats to the organizers and Mireia, particularly. So I have two broad questions. One is you showed very nicely the oral microbiome. It's pretty much similar. I mean, I think I don't remember exactly the ratio, but it's one of the highest values that you found to be more, let's say, present in more than one individual. So I would just like to ask you, what are your thoughts in sexual relationships? Is this really a direct factor? And also how we could distinct these factors from, for example, households that eat similarly diets, is somehow that we could stratify this. My second question, if you can.
Maybe,
just go ahead.
I'll answer first these two, and then I can continue with the other ones you might have. So you were asking first, on the high transmission of the oral microbiome. Indeed, to us, this makes sense. First, because of the properties of bacteria in the mouth. So they are tolerant in this case. They can survive in the environment. So it makes sense that it's easier for them to be transmitted than those in the gut. Also, oral bacteria have saliva as a transmission vehicle. And indeed, we see that partners share more of their mouth bacteria than them with the offspring. Then, of course, our study is computational. So what we did was comparing metagenomes. So we don't know exactly how transmission routes were. But yeah, we could think that any kind of close contact would influence microbiome transmission. That was the first question. Then you also asked
the diet.
The diet. Yeah,
factor.
of course. Yeah, perfect. Indeed. So if we look at microbiome composition at the genus or at the species level, this could, and when we see similarities there, that could very well be due to similarities in lifestyle. So diet is a very clear one. But also disease and so on. And so that's why it was important to do this study at the strain level after seeing that strains were so personal. So we did some tests, and these are in the supplementary materials of the paper, where we are seeing that two different individuals that are not in contact, it's very unlikely that they share the same strain. So if that does not happen, if individuals are not in contact, like here you can see the 0% as a median, then when we see this higher strain sharing, we can infer that there was a transmission event. So that's why it's important to do strain level analysis to this to answer these kind of questions. You had a second question, right?
Yes, I do, but I also think there's another one in the comments. Maybe I just wait a bit, then in case I can ask you another one. Yeah, thank you so much.
Thank you.
So yeah, there is one in the chat. So from Eugenio, do pathogenic bacteria share the same mechanisms of transmission as commensal bacteria?
That's a very interesting question, and one for which we don't have an answer yet. So we don't know exactly the mechanisms of transmission of the gut and the oral microbiomes yet. It seemed that pathogenic bacteria have more aggressive colonization strategies. So they could be exploiting slightly different mechanisms, but this is something that we are going to look at in follow-up research. We don't know it yet.
I also had a question regarding, I don't know, nowadays we hear a lot, right, that there is a lot of antibiotic resistance. Is there any correlation, or did you check any correlation with different bacteria species with resistance to antibiotics?
Yeah, that's also a very important point, because I mean, we can see indeed that antibiotics kill many of the bacteria, the health-associated bacteria in our microbiomes, and some are acquiring these resistances. Here at this larger scale, we did not really check for the transmission of antibiotic-resistant genes. In the smaller one, the one in multiple generations, we did, and we could not really see direct transmission. But there, the methods still need to improve a bit. So that could also be a possibility, yes.
Okay, thank you very much. Romulo, if you want, you can ask her.